Good afternoon, ladies and gentlemen, and welcome to the annual William E. Paul Lecture as part of the NIH Director's Wednesday afternoon lecture series. This is the first lecture in this newly named series dedicated to the memory of the former chief of the Laboratory of Immunology at NIAID, Dr. William E. Paul. We are very pleased that Bill's wife, Marilyn, other members of Bill's family, as well as many of Bill's mentees and long-term colleagues are joining us for today's special lecture. Bill first joined the Institute in 1968, along with Nobel laureate Baruch Benacerraf. I remember Bill's arrival very well, since this was the year that I began my own immunology infectious disease fellowship here at NIAID. Bill was 17 years old, and I was 14 years old. He was very bright and was appointed the chief of the Laboratory of Immunology 2 years later in 1970, a position that Bill held for more than 45 years. Under Bill's leadership, the Laboratory of Immunology became one of the premier centers of immunological research in the world. His research resulted in many seminal discoveries that advanced the fields of immunology and provided the foundation for our current understanding of T-cell and cytokine biology and their roles in host immunity and disease. Bill discovered interleukin-4 and delineated its role in allergic and inflammatory diseases. He also delineated the fundamental mechanisms by which T-cells develop into mature effector cells that are responsible for key activities in the areas of infection, inflammation, allergy, autoimmunity, and asthma. From 1994 to 1997, Bill also served as the NIH Associate Director for AIDS Research and the Director of the Office of AIDS Research. During his tenure, he led the development of the first strategic plan and unified budget for all NIH-sponsored HIV/AIDS research programs and activities. The first comprehensive evaluation of the NIH HIV/AIDS portfolio was conducted during the time that Bill was the OAR director. This review resulted in a refocusing on basic immunology, as you might expect, and vaccines within the NIH HIV/AIDS research program. Bill was a mentor to many researchers who went on to become leaders in the field of immunology. His many awards honors include the Max Delbruck Medal, the Clemens von Prickett Medal, as well as the Lifetime Achievement Award and the Excellence in Mentoring Award from the American Association of Immunologists. We still feel Bill's loss today. To honor and remember the legacy of this very special scientist and person, the WALS lecture today is dedicated to his honor. Before I introduce our speaker, I'd ask Marilyn Paul to please come up and say a few words.
I want to thank the Director of NIH, Dr. Francis Collins, and Dr. Anthony Fauci, Dr. Michael Gottesman, and Dr. Pamela Schwartzberg. But most of all, I want to thank the Immunology Interest Group Steering Committee, who came up with the idea of naming a Wednesday afternoon lecture in memory of Bill. Now, this is not the first time I've come to a Wednesday afternoon lecture. I can still remember when it was in 1982, as a very young scientist, Bill was selected to give the Wednesday afternoon G. Burroughs Mida lecture. He was so surprised and delighted to be recognized in this way so early in his career. And I came, and I sat beaming in the first row, and I can even remember the dress I was wearing. Bill and I are both here again today, but in very different roles. Bill is here only as a memory, as an esteemed alum of the Laboratory of Immunology, the NIAID, and the NIH, and as father of the Vaccine Research Center. Bill was proud to be a member of the generation of scientists who opened the doors to immunology to its power and potential. And I am here today to thank the NIH for affording Bill an extraordinary career. Our family had the pleasure of living with a man who enjoyed every day that he went to work. Bill's success brought joy to our home and gave me and our family a wonderful life. And now, my family and I appreciate the honors being bestowed on Bill's memory. We will always be grateful to Bill's years at the NIH. Thank you for coming here this afternoon.
Thank you very much, Marilyn. It is now my great pleasure to introduce today's lecturer, Dr. Thomas A. Waldman. In Tom's presentation today, he will share his perspectives on the IL-15 Janus Kinase-Stat system and its implications for immunotherapy of autoimmune diseases and cancer. Tom received his medical degree from Harvard Medical School and joined the NCI in 1956. He has served as the chief of the lymphoid malignancies branch, which was formerly the metabolism branch, since 1973. His research focuses on the IL-2 receptor system and the growth of both neoplastic and normal cells. He defined the IL-2 receptor alpha and beta subunits using the now FDA-approved daclizumab antibody that he himself discovered. Tom also co-discovered IL-15 and performed the first in-human clinical trial of this agent in patients with metastatic malignancy. His research has defined molecular abnormalities of the common gamma cytokine, JAK-STAT signaling pathway in HTLV1-associated T-cell lymphoma, and he conducted a clinical trial of JAK inhibitor in patients with disorder. Tom's laboratory has also focused on studies of T-cell receptor and immunoglobulin genes for the diagnosis and monitoring of therapies for lymphomas and leukemias, including production of antibodies to the IL-2 receptor and the use of the IL-2 receptor-directed therapy. Tom's lab continues today in making major scientific contributions in cytokine biology and immunology that are significantly benefiting the areas of autoimmune diseases, cancer, and organ transplantation. So please join me in welcoming my friend and colleague, Dr. Tom Walmann, as he presents his talk entitled The Two Faces of the IL-15 Janus Kinase-Stat System, Implications for the Immunotherapy of Autoimmune Diseases and Cancer.
Well, thank you, Tony. It's a true pleasure and a great honor to have the opportunity to present the inaugural William E. Paul Honorary Lecture. Bill Paul is a legendary figure in the field of immunology. Bill has been an inspiration and my treasured friends for 55 years, since 1962, when he joined the MC Lee and Roy Hertz for a stint in the endocrinology branch of the NCI, where he treated women with choriocarcinoma with the drug methotrexate. However, he soon caught the immunology wave. Bill Paul is the shining icon and pioneering giant of contemporary immunology. He had an encyclopedic knowledge of the field. For 3 decades, he was chief editor of the Annual Review of Immunology, which under his guidance for 7 years was the most cited publication in biomedical science. He initiated and edited Fundamental Immunology, the most definitive reference in the field. He was, for a period, the editor of the journal Immunity. He was president of the American Association of Immunologists, the American Society for Clinical Investigation. Furthermore, he was chief of the Laboratory of Immunology from 1970 until his death. With his persistently positive attitude and his generosity, the laboratory was and is a resounding success. We all appreciated his ability to ask the most probing questions, yet make us feel we were receiving an accolade, a reflection of his modesty about his remarkable intellect. I and many of you in the audience have benefited from his generosity by being nominated for an honor, award, or a membership in an august society, actions where he appeared to have more enjoyment from our successes than from his own accolades. Bill's contributions to the research in the field of immunology are extraordinary. One of his first and important works was with the future Nobel laureate, Dr. Baruch Benacerraf and Ethan Shivak, Ira Green, and Alan Rosenthal, who showed that IR genes control the capacity of simple carrier molecules, such as mono and diamino polymers, to elicit immune responses to any haptenes conjugated to these carriers. He showed that MHC class II molecules played a critical role in the presentation of antigens by APCs to T cells, and that polymorphisms in class II determined the specificity of such control. His more recent seminal contribution with Maureen Howard was the discovery of IL-4, the elucidation of the molecular basis of IL-4 signaling, dissection of molecular genetic control of the expression of IL-4 and other Th2 cytokines, and the delineation of the general principle of positive cytokine feedback regulation of CD4 T-cell effector polarization. For 4 decades, it has been my privilege to share an interest in the common gamma cytokines with Bill. Over the past 35 years, a major focus of my laboratory has been on the roles of IL-2 and IL-15 in the life and death of lymphocytes and the implication of these observations for the immunotherapy of cancer. I have only a moment to touch upon IL-2, where with Takashi Uchiyama, we produced anti-TAC, daclizumab, Zimbrita, a monoclonal antibody that with Warren Leonard and Warner Green was used to define and molecular clone the gene for IL-2 alpha, the discovery of CD25. Through the decades, we have developed this antibody where in its humanized form for therapy, it was approved by the FDA for the prevention of organ allograft rejection and for the treatment of patients with multiple sclerosis. However, today I will focus on IL-15, a cytokine pivotal in the maintenance of NK cells and memory phenotype CD-T cells. There will be 2 parts to my talk. The first, familiar to many of you, will be our efforts to bring to fruition IL-15 in combination therapy as an effective approach to the treatment of metastatic malignancy. The second part, which I have never presented, will be on disorders of IL-15 and its signaling JAK-STAT system in autoimmune diseases and T-cell malignancies, and our efforts to develop agents to take that into account and treat these disorders. So in terms of Janus, the Roman god, he looks both backwards and forwards into the future as I will. And I will also give him both good and evil faces. IL-15 useful in therapy, IL-15 involved in disease pathogenesis. So IL-15, as shown here, is involved in an immunological synapse where antigen-presenting cells and IL-15 or alpha present its always partner, IL-15 in yellow, to cells in the beta and gamma chain of its tripartite receptor. I will tell you, IL-15, when given to people, has the capacity to dramatically increase the number and activation status of NK cells, a 30-fold increase in the number of conventional NK cells, a more than and in rhesus macaques to increase the number of CD8 cells 100-fold to 100 per cubic millimeter. However, in each case, there is an induction of a checkpoint. Thus, in order for IL-15 to reach its potential, it will have to be used in combination therapy that circumvents these checkpoints, that is rational, that is functioning in animal models, and is just now being translated into clinical trials. So IL-15, which as usual we discovered in serendipity while studying IL-2 receptor, is a 14-15 kilodalton member of the 4-alpha helix bundle cytokine family. It and IL-2 share much. They both have as their receptors a heterotrimeric one with the gamma chain also used by IL-4, 7, 9, and 21. And they share a beta chain, CD122, which I've named here IL-2, IL-15, R-beta. Furthermore, each has its unique alpha chain that for IL-15 is really a pivotal partner. They signal through JAK1, JAK3, and STAT5. So for a long time, one thought IL-15 must be IL-2 light. They must be very redundant. And in part, that is true. However, they do have really unique roles. IL-2 has a role in preventing immunity to oneself, to prevent autoimmunity, and is pivotally involved in the fitness of regulatory Tregs and in the phenomenon of activation-induced cell death. In contrast, IL-15 inhibits IL-2-mediated activation-induced cell death, and its role in terms of long-term protection against invading pathogens stimulates the development of CD8 memory phenotype T cells. Knockouts of these IL-2 and IL-2 alpha versus IL-15 and IL-15 or alpha have quite distinct phenotypes as predicted by these different roles. With IL-2 knockouts, there's massive enlargement of peripheral lymphoid organs as one loses checkpoints, high levels of immunoglobulin subclasses, and autoimmune diseases. In contrast, with IL-15 or the knockout of its private receptor, IL-15 or alpha, there is no such lymphoid enlargement, autoimmune disease, or impaired or activation-induced cell death. Rather, there is marked reduction in the number of NK cells, NKT cells, gamma delta cells, interepithelial lymphocytes, memory phenotype CD8 T cells, cells that depend on IL-15 for their maintenance and production. So how can there be difference? There are many differences, but I will only show you one developed by Sigrid Dubois in the laboratory. IL-2 is a secreted molecule that acts on preformed high-affinity heterotrimeric receptors, usually on other cells. In contrast, IL-15 is predominantly not secreted, but rather acts as part of an immunological synapse. IL-15 receptor alpha on antigen-presenting cells, indeed IL-15 and IL-15 or alpha, as shown on your left, are made together when the cell is exposed to interferon, CD40 ligation, or toll-like receptors. And these cells with recycling IL-15, IL-15 or alpha circulate for a number of days until they meet with their target, a beta-gamma expressing NK or CD8 T cells that does not express IL-15 or alpha and transpresents IL-15 signal to the new cell for its benefit. Well, we studied IL-15 due to the differences we've just discussed in a considerable number of tumor models. This was the first study that we did in the CT26 tumor model, and one sees a modest effect of IL-15 prolonging the survival of tumor-bearing mice. Studies of these 4 different types of studies such as these, and the indication that it was not really very toxic in rhesus macaques, led the immunotherapy agent workshop, which identified agents that might be of value in the clinic. IL-15 was number 1 in its list of such potential useful immunotherapeutic agents. With that in mind, and with the financial help of our scientific and institute director, we had an opportunity to oversee the biopharmaceutical development program, Drs. Jovandich and Creekmore, in the production of E. coli IL-15 for human use. And with this, we, with Kevin Conlon, performed the first in-human study of IL-15 in patients with metastatic malignancy. The patients had a much more toxic path than the rhesus macaques. There was fever, shaking chills, hypotension, and one had to de-escalate with the dose of IL-15 to 0.3 of a microgram per kilogram. Nevertheless, as shown here, following the administration of IL-15 by bolus infusion, there was an immediate virtual disappearance of NK cells from the circulation that may be marginated in postcapillary venules or actually enter the tissues. But before Ki-67 new synthesis kicks in, the NK cells began to reappear in the circulation so that one saw by bolus infusion increase in the number of NK cells. However, we were not satisfied, especially because there were no PRs or CRs. That the 5 signal lesions had a 20 to 30% reduction in size. In 2 patients, there was a disappearance of lung lesions. We noted this high Cmax, the high level of IL-15 if it was given by bolus injection. When this was done in rhesus macaques, as shown here, with 20 micrograms per kilogram, the Cmax was 720 nanograms per mil. We reasoned if we had a different dosing strategy, we might have lower Cmax and less toxicity and more efficacy, and that was the case. When we gave IL-15 subcutaneously, the Cmax was lowered by a log, and yet another log if by continuous intravenous infusion, where one had a much longer period of time when the recipient had an active dose of IL-15. So with the Cancer Immunotherapy Network, we did a study of 19 patients with IL-15 in a dose escalation trial. And one noted that in the outpatient setting, this was quite well tolerated with a grade 2 pancreatitis as the only serious toxicity. One noted an increase in the number of NK cells, especially in the CD56 bright NK cells. Furthermore, we gave the IL-15, it's shown at the base of each of the graphs in light blue, but the maximum response in NK cells appeared after we had stopped giving the sub-Q IL-15. This was more dramatically seen when we administered with Kevin Conlon IL-15 by continuous intravenous infusion to patients with malignancy. Here, the IL-15 was administered during the red arrows, and there was a burst of lymphocytes that appeared at the time, next few days following termination of the infusion. During the rapid proliferation, perhaps cells were not returning to the circulation or were in post-capillary venules. But anyway, one noted this marked increase in the number of NK cells 30-fold increase in NK, 350 CD56 bright NK. To remind you that there are two types of NK cells. The majority involved in cytotoxicity are CD56 dim in blue. CD56 bright, the ones most stimulated by IL-15 were there. They had been called immunoregulatory, producing IL-10, and were not felt to be very effective cytotoxic cells. However, Sigrid Dubois has demonstrated in green, that in red, that the injection of IL-15 was associated with an increase in both 56 bright and 56 dim cytotoxicity, and this cytotoxicity included that mediated by NKp46, by ADCC with an anti-CD20 monoclonal with CD16, and with NKG2D MICA systems. So we are generating cytotoxic cells. So if we're generating this huge number of cytotoxic cells, why are we not getting efficacy? Well, as you know, NK cells of a person are inhibited when they recognize self-class 1 MHC expressed on the tumor. So if it's expressed as HLA-E, NKG2A is the receptor of this negative signal. If it's class 1A HLA, then cares for humans. So we're having a turnoff of the system. And although, so that we have to use IL-15 in combination to get efficacy, and we do this by taking advantage of the role of NK cells in the phenomenon of antibody-dependent cell-mediated cytotoxicity killing of tumor cells. And so here is one of many different types of studies. In a fully immunocompetent mouse, EO4 leukemic cells of the mouse were transfected with human CD20, and the mice were treated with the anti-CD20 monoclonal antibody rituximab. When we did this in mice, in red, IL-15, very modest effect, rituximab, moderate. But the two together universally cured 80 to 100% of the mice in various studies. In a xenograft model, alemtuzumab, anti-CD52, in a model of HTLV-1-associated adult T-cell leukemia, a major focus of the laboratory, we again saw in yellow on the left little effect with IL-15, Campath, anti-CD52, modest, and the two always giving you an additive synergistic effect. If we did these studies in Fc receptor gamma knockout mice, those that cannot do ADCC, all efficacy was lost. So we are looking into what cell is involved. Most people believe ADCC is mediated by NK cells, but there's a group headed by Jeff Ravitch at Rockefeller that concludes that no, it is macrophages that is involved in the ADCC killing. So we did our EL4 CD20 study, and we eliminated NK cells with acylo-GM1 in red and lost all biological efficacy. But also, when we used chlordonate liposomes, as shown in blue, we lost the majority of activity. It appears one needs both cells acting in combination. And we have shown with deletion studies and with IL-15 that one must have both cells and IL-15, which induces RAY1 or MICA for humans and NKG2D, the latter on NK cells. One induces on NK cells more CD16, more Fc receptor gamma 4 needed to bind the monoclonal. So we are augmenting with IL-15 both the number and the functional capacity of NK cells. With these in mind, we're initiating a series of studies where we're using anti-cancer monoclonal antibodies in association with IL-15. Some of these are in adult T-cell leukemia lymphoma, which is an aggressive disorder of human CD3, CD4, CD25 cells, which we in 1984 showed to be profound immunosuppressors. They are leukemias of Tregs. And these, there is no curative therapy other than bone marrow transplant. So when in the past we studied alemtuzumab, anti-CD52, we noted that 12 of 15 patients with the acute leukemic form of this disease had a partial or complete response, and 2 of 3 chronic. However, the responses were of short duration. We wish to increase the proportion of responders, but especially the duration, by adding IL-15 to augment the ADCC of alemtuzumab. So we start by treating in a dose escalation form with IL-15, followed by alemtuzumab in a conventional dosing strategy. We've only treated 2 patients, the first one with the acute form of ATL. I show here where in blue are the number of cells, lymphocytes in the circulation, and in red, IL-2 alpha, a mediator, surrogate marker of tumor activity. One thus is having in the periphery a good response. But we also note the limitation of this approach. The antibody does not pass the blood-brain barrier, and thus this patient has developed neurological adult T-cell leukemia, which for the future we will have to evaluate that beforehand and use intrathecal methotrexate. But anyway, this is one. We are approaching an effort to use avelumab, an anti-PD-L1 antibody that does ADCC, in the treatment of T-cell malignancy and one form of solid tumor. And similarly, use this basic approach, IL-15 with an anti-cancer monoclonal antibody, with the anti-CD20 obinutuzumab for CLL. So we are using in refractory and relapsed CLL anti-CD20 monoclonal antibody plus IL-15. It is hoped that we can thus take advantage of the effect of IL-15 on macrophages and especially NK cells with this approach. However, IL-15 always has a partner, IL-15 or alpha. Indeed, there are those who consider these a heterodimeric cytokine. What was noted by others is that they must be together in mice to generate effective IL-15. If one has an animal with bone marrow transplants of cells that express only IL-15, and in the same animal, only IL-15 or alpha, as shown on your left, one does not generate effective IL-15, and the mouse has no NK cells which depend on IL-15 for their production. In contrast, if they are produced by the same cell as shown on the right, you have IL-15 effectively produced, transpresented, and NK cells developed and are maintained. In addition, there were studies of Coca and all, where normal NK cells were injected into wild-type mice in blue or to mice missing IL-15 or alpha. And as you note, in wild-type mice, there's a meaningful survival, but virtually no survival in mice who do not have IL-15 or alpha. Thus, one is moving to situations where one will administer not IL-15, but IL-15 in combination with IL-15 or alpha, generated by George Pavlakis in the upper right, And if you note the pharmacokinetics of that complex in blue in the lower left, it is better than just plain IL-15 shown in red, a longer survival. And in the lower right, it is more effective in murine models of tumor than IL-15. Longer survival and more effective. A third arena involves checkpoints. Here, IL-15 leads to the production of IL-10, of PD-1 on lymphocytes, and on TIM and other checkpoints. And so we looked at the combination of IL-15 with anti-CTLA-4 alone, anti-PD-L1 alone, or the triple combination. Here, in this TRAMP-C2 prostatic cancer model, we see little effect of IL-15 that is not augmented when we add either anti-PD-L1 or anti-CTLA-4 alone. But when all three are added together in green, we see additivity or synergy. We are translating this into a clinical trial involving this triple combination for patients with solid tumors that are refractory and relapsed for treatment. In the final of these groups, we note that in rhesus macaques, if we give IL-15 by continuous intravenous infusion, as shown in red at 20 micrograms per kilogram per day, there is a 100-fold increase in the number of such cells to 80 to 100,000 per cubic millimeter. In humans, the response is considerably less, but nevertheless, we wish to take advantage of the CD8 cells generated and that are activated. Yet again, we come upon a checkpoint. That is, if you give a gamma cytokine out of sequence, normally you would give an antigen, a co-stimulant molecule, and then the cytokine. But here we're giving the cytokine first. And when that happens, as Sikosil showed initially, one has a failure of CD4 cells due to upregulation within the cell of CIS, and the checkpoint SOX3. So these two agents dephosphorylate the signaling pathway, and you lose CD4 cells. As a consequence of loss of CD4 helper cells, there are the generation of helpless CD8 T cells, which intrinsically are not hurt by the IL-15, with impaired specific immunity toward novel tumor antigens. Ligation of CD40 reactivates antigen-specific T cell responses. Already in 1998, in 3 back-to-back papers in Nature, the way that lymphocytes, antigen-presenting cells work, and the role of CD40 were defined. Initially, helper cells interact with the presenting cells. The helper cells then can leave, and CD8 cytotoxic cells now bind to the presenting cell and function. But in the absence of the helper cell, you have, as I've noted, helpless CD8 cells. But these lack of CD4 cells can be reversed if one has an agonist to CD40. We had shown this already. The combination of agonistic anti-CD40 monoclonal antibody and IL-15 provided effective therapy for an established 100 millimeter subcutaneous TRAMP-C prostatic cancer in mice. Again, IL-15 very modest, anti-CD40 moderate, but always 80 to 100% of the animals are cured with the combination of anti-CD40 and IL-15. We looked at the generation of tumor-specific CD8 cells using the tetramer agent provided by Jim Allison. You note that on your right, in yellow, that IL-15 alone did not augment, anti-CD40 did not augment, but the combination markedly augmented the number of these cells that were involved in the anti-tumor activity. So we are joining with Jeff Ravitch to use an optimized anti-CD40 monoclonal antibody plus IL-15 for patients with metastatic malignancy. So in summary of the first part, IL-2 and IL-15 have contrasting roles in the life and death of lymphocytes. IL-15 or alpha, IL-15 recycles and presents IL-15 in trans to neighboring NK and CD8 T cells. IL-15 is a critical factor in the function and memory of NK cells and memory CD8 T cells. The optimal use may require co-administration of agents such as anti-CD40 to induce the expression of IL-15 or alpha. We have completed bolus subcutaneous continuous infusion of IL-15 and have seen marked effects on the number and state of activation of NK cells. With the combination of IL-15 and IL-15R alpha versus IL-15 alone had a greater effect on NK stimulating activity. The administration of IL-15 with a combination of checkpoints, anti-CTLA-4 and anti-PD-L1, was associated with anti-tumor synergy. In addition, IL-15 increased ADCC and anti-tumor efficacy of cancer-directed monoclonal antibodies, a major program now in effect. And the combination of IL-15 with an agonistic anti-CD40 monoclonal yielded augmented tumor antigen-specific CD8 T cells and anti-tumor efficacy. So far, I have indicated where IL-15 may be of value. We also have used it with lineage Pereira as a constituent of vaccines directed toward infectious agents, toward tumors, and toward bioterrorism agents. But now I'm going to change gears entirely and indicate that IL-15 plays a pathogenic role in a number of tissue, organ-specific autoimmune diseases, and it, and especially its pathway, is activated in an array of T-cell malignancies. And we'll give you first clue on a number of agents that have been used to address these issues, agents that have worked with cell lines, with ex vivo proliferating human tumor cells, and in mouse models, and that have been initiated in early clinical trials. So a concern with IL-15 is it's an inflammatory cytokine that induces the expression of IL-6, interferon gamma, TNF-alpha, and IL-1 beta. It inhibits the self-tolerance mediated by IL-2-induced activation-induced cell death. It facilitates CD memory T-cell survival, including cells directed against self. Thus, if it were indiscriminately expressed, it would lead to inflammatory autoimmune diseases. Diseases that go beyond just demonstrating high IL-15 levels are shown on this slide. Refractory celiac disease, alopecia areata, vitiligo, and type 1 diabetes. In most of these, the effector agent is an NKG2D CD8 T cell. But it is also present as an issue in a number of T-cell viral-induced diseases. The retrovirus, human T-cell lymphotrophic virus 1, transactivates IL-15 and IL-15 or alpha, and thus the disease HAM-TSP, a neurological disease like multiple sclerosis, and the leukemia, adult T-cell leukemia, have a IL-15 component. Thus, we have wish to target IL-15, to prevent it interacting with its receptor, to prevent its signaling pathway as an approach to these disorders. Indeed, for example, I won't show data, but in alopecia areata, if you obstruct the signaling by IL-15 in 7 of 11 such individuals, there was a major return of hair growth. So again, I mentioned to you that IL-15 is transpresented to beta and gamma expressing cells. And many of the initial approaches involve agents that block this interaction, either small molecules binding solely to beta or solely to gamma to prevent heterodimerization and activation of JAK-STAT, or a monoclonal antibody that I now introduce, humic beta-1 in humans, TM beta-1 in mice. And this binds to the IL-2, IL-15R beta shown in blue and blocks IL-15 transpresentation. So humic beta-1 inhibits IL-15 transpresentation and its action on T and NK cells. It has prolonged cardiac allograft survival. Trials are underway in ham-TSP, in refractory celiac disease, and were completed just for toxicity in LGL leukemia. So let us turn to some of the autoimmune disorders. We reasoned that if IL-15 and IL-15R alpha were incorrectly expressed by tissues, and I mentioned that IL-15 message is available in many epithelial tissues in addition to monocytes and macrophages. We reasoned if they were expressed, this would draw NK cells, CD8 cells, and monocytes to the tissues and lead to autoimmunity. So we developed an assay for IL-15R alpha, IL-15. And as you see here, the levels in the serum were elevated in celiac disease. In this enormously oversimplified diagram from Bana Jabri, among many others, one has for patients that are DQ2 or DQ8, that is genetically susceptible, seeing the stress of gluten, one induces in the epithelial cells, on the one hand, IL-15 and IL-15R alpha, which is transpresented to CD8 cells, activating them. Also, IL-15 activates NKG2D expression by these CD8 cells and MIC-A production by the epithelial cells. So at multiple levels, IL-15 increases the events leading to tissue destruction in celiac disease. And so in a number of models, the implication that IL-15 plays a significant role have been underlined. Here in IL-15 transgenic mice that express human IL-15 in intestinal cells using an enterocyte-specific promoter to drive the transgene, developed, as shown in the centered panel, florid inflammation of the duodenal region and villus atrophy. When we use TM beta-1, the equivalent of humic beta-1 from mice, that interacts with CD122, IL-2, IL-15R beta, and blocks IL-15 action, it, as shown on the right, reversed all of the effects. This was started 4 months after the animals were born, 2 months before this picture. So one is able to completely reverse the macroscopic and microscopic aspects that look like celiac disease by blocking IL-15 binding to its receptor. One also sees such an event in patients with type 1 diabetes. Here we're again measuring IL-15R alpha, IL-15 in green on the right, showing elevated levels compared to the very low levels in normal serum. And to that end, we generated IL-15, IL-15R alpha, and the combination transgenic mice expressed on an insulin promoter, thus in the beta cells of the islets of the pancreas, shown at the bottom for their expression. These double transgenic mice expressing on their beta cells IL-15 and IL-15R alpha early developed hyperglycemia and loss of these cells, paralleling the observation in type 1 diabetes. If one inhibits this signaling, either on the left with TM-beta-1, which you'll remember blocks IL-15 binding to its receptor, as shown in red, we reverse the hyperglycemia. And tafasitamab, in its early name, with JAK1/2/3 inhibitor, as shown on your right, again, reverses the hyperglycemia that had been present. So if you block IL-15, you reverse the diabetes in it. And more physiological or pathophysiological, we turn to patient material from the Pancreas sharing group. And you will note at the top in A, we are immunochemistry staining for IL-15 in pancreatic tissues from a patient with type 1 diabetes. In all cases, IL-15 could be demonstrated in these islets of the patient. Below that, we used microdissection of the islets and nanostring analysis of the expression of message for IL-15 in B and IL-15 or alpha in C. And you'll note the higher than levels in the patients with type 1 diabetes and high serum levels of soluble IL-15 or alpha. So a hypothesis is that a Coxsackie enteroviral infection that has been shown to be associated with diabetes by some occurs first, that this induces interferon, and interferon is the greatest inducer of IL-15 and IL-15 or alpha, which then brings to this tissue macrophages, NK and T cells, which destroy the beta cell. In any case, one may think about the possibility during the early phases of type 1 diabetes, before there's full destruction of the beta cells, of inhibiting IL-15 action. But I will now turn to T-cell lymphoma and tell you that there is pervasive activation of what really should state the gamma cytokine receptor JAK-STAT pathway, and in T-cell lymphoma. That is, all of the different types of T-cell lymphoma that have been examined for activation of the pathway have shown from 10 to 86% of the patients with that subset as bearing an activation of this gamma cytokine JAK-STAT pathway. And the reason I will say it that way is I'm going to have to go more rapidly through summaries, but to state that you need the whole pathway. It doesn't matter if you have a mutation of STAT3 or JAK1. You need the whole pathway, not just the so-called activating mutation, with the exception of ALK-positive cells. So the common gamma cytokines, as we say, signal through JAK1 and JAK3, STAT3 and STAT5 for T-cell malignancies. This activation is pervasive as measured by phosphorylation of STAT3 and STAT5 in the nucleus. That is what is the most common finding. The mutations are a mutation here, a mutation there, but what is universal for the activation of the pathway is phosphostat-3 and 5 in the nucleus. RNA loss of function shows that one needs STAT and JAKs, whether or not they are mutated. And these mutations are not sufficient to support leukemic cell proliferation, but only augment signals from upstream in this pathway. In addition to mutations, there are many abnormalities, increased cytokine production, for example. And they're activating mutations. There was a requirement for a functional receptor. Whether the mutations were a JAK, they need to have a functional cytokine receptor as a scaffold for cross-activation of JAK kinases and as a docking site for recruitment for STAT transcription factors. So on the basis of these insights, JAK kinase inhibitors are being initiated for the treatment of T-cell leukemias and lymphomas. Here is just one example of 7 cell lines. 6 of these express activation of STAT3. These are ALK-negative anaplastic large cell lymphoma that are not receiving cytokines from the outside, but they're manifesting for an array of reasons activation of this pathway in a way that is responsive and is inhibited by JAK inhibitors. I just show you this to show that there are a whole array in the colors at the bottom of different subsets of T-cell malignancy, but that each of these manifests STAT mutations predominantly in the SH2 domain. If you use shRNA deleting, targeting STAT3, you will induce cell death if there is a phospho-STAT3 cell in red, but not if it's a phospho-STAT3 positive ALK-negative cell line, whether or not they're mutated. So if you have activation of the pathway and you inhibit STAT3, the cell lines die. But even upstream of these STAT mutations, you need JAK1. So if you eliminate JAK1 with shRNA, you again have cell death. So as I will say, you need the whole pathway no matter where the mutation is. And what is happening is you are increasing the binding of the STATs to each other. They're homodimers of phosphostat. And when you have a mutation, these two come together more readily. That is, they respond to a very low dose of IL-2 that wild-type STATs would not, and they respond for a longer period of time. This latter is shown here in the middle 2 and bottom 2 panels. In the middle 2, you will note that with wild-type STAT5b, if you give IL-2 and then withdraw it and look for how long the signal lasts, it lasts for 1 hour. If you do this with the mutant, that same signal is still active at 6 hours. So you have a change in the binding of these STATs with each other, a change in the responsiveness to upstream signals. And this is shown here on the black and green arrows at the bottom of the slide, that you're increasing the response to upstream signals. STAT mutations are also very common. JAK mutations are all very common in T-cell malignancies, predominantly in the pseudokinase and kinase domains. But again, as I've noted before, they are not enough. One needs to have a functional cytokine receptor. And this is shown here in the work of Liu et al. If you look at the panel on the top and the first 4 segments, in the second segment from the left, where you have mutant JAK2, you do not have phosphostat-5. In the third panel, where you have the erythropoietin receptor, you do not have phosphostat-5. But where the two are there together, where the mutant JAK is associated with the cytokine receptor, now there is signaling in the pathway in this and in many other systems. So now we have generated, you need the STATs, you need the JAKs, and you need the receptor. In the case of adult T-cell leukemia, you need the gamma cytokine at the top as well. Adult T-cell leukemia is a leukemia of CD3, 25 cells that we showed profoundly immunosuppressing, and is a leukemia of Tregs. So here, HTLV1, this retrovirus, generates TACs, transactivates IL-2 and the IL-2 receptor, IL-15, the IL-15 receptor, and IL-9. And so these cells, when taken out of the body, peripheral blood mononuclear cells, the leukemic cells proliferate spontaneously without added activators. And this can be inhibited in many ways. In one way, BNZ1 is a peptide generated by ex-postdocs, Nazli Azimi and Yataka Tagaya, from the delta chain of IL-2 and IL-15 so that it binds tightly to the gamma chain, but not at all to the beta chain. And so it enters there, it prevents the wild-type cytokine entering IL-2 and IL-15 and prevents the heterodimerization needed to signal it. Similarly, from Garcia and Leonard, one has H2RH9, RETR, where one has a modified IL-2 that binds tightly to the beta chain and not to the gamma chain, a gamma with a similar effect. So these small molecules are able to block simultaneously IL-2 and IL-15. BNZ1 is in the clinic in normal volunteers and leads to, as expected, profound reduction in NK cells and other events. However, one can look at the ex vivo proliferation, the study peripheral mononuclear cells in 6-day culture, and look at the proliferation of the leukemic cells. In red, you will note that if you give an antibody, daclizumab, to IL-2R alpha and to IL-9 and IL-15, one inhibits this proliferation because you inhibit the two autocrine-paracrine loops. With ruxolitinib, a JAK1/2, I emphasize 2, inhibitor, one has a similar effect. And one could show efficacy in animal models where we gave the ruxolitinib by continuous pump. So on the basis of these insights, we have a clinical trial of ruxolitinib in patients with smoldering and chronic adult T-cell leukemia who have that spontaneous proliferation inhibited ex vivo. Although this agent was very effective on ex vivo cells, it was disappointing in these studies. And we feel the reason for this lies in the pharmacokinetics and pharmacodynamics of ruxolitinib as given as approved by the FDA, which is, let's say, 20 milligrams every 12 hours. And it has a short pharmacokinetics and dynamics. Here in Sigrid Dubois' data, we show you in the left what happens to phosphostat that is constitutively expressed in the ATL cells. We will note at 0 hour in red, the cells are phosphostat positive. When the patient received the ruxolitinib, at 1 hour in green and somewhat at 3 hours in orange, one had inhibition of phosphostat. But for the majority of the time between doses, the pathway was active and not inhibited. And this reflects the off-target JAK2 inhibition of ruxolitinib. That is, it inhibits JAK2 not used by T-cell leukemia cells and is unwanted. JAK2 is used by thrombopoietin, and therefore there's great fear of thrombocytopenia, and so it is inadequately working in this system. And since you don't need it, we are looking at a great number of other alternatives, JAK1 specific inhibitor, JAK3 specific inhibitors, and looking for inhibitors with the pharmacokinetics of ibrutinib, that is, long-term effects on the system. If you have a pure JAK1 or JAK3 inhibitor, you should inhibit the actions of IL-2 and IL-15 that use this system, but you should not inhibit the action of IL-3 that works through JAK2 alone. So on the upper, we have a cell line, 32D beta, that responds to all of these cytokines. And you will note that as it should, each of these JAK1 and JAK2 inhibitors inhibits IL-2 or IL-15 stimulated cells. On the bottom, they do not inhibit IL-3 stimulated cells. They do not inhibit JAK2. However, ruxolitinib does. And so we're developing a strategy that utilizes a JAK3 inhibition as one of the approaches, one of the elements of multi-drug treatment of ATL smoldering and chronic patients. Here, with certain of the cell lines are responsive to this inhibitor. So, in summary, there are many different agents that attack this pathway at different positions and that could be useful in the treatment of organ-specific autoimmune diseases we've discussed and T-cell malignancies where there is STAT phosphorylated in the nucleus. There are antibodies to IL-2 and IL-15. The latter are in the clinic and are being used in celiac disease. There are antibodies to the cytokine receptors, or daclizumab, approved for transplantation and multiple sclerosis. There are the small molecule inhibitors, H9RETR, that binds to the beta chain but not gamma, or BNZ1 that acts on the gamma constant but not the beta chain. These two preventing homodimerization of the receptor, signaling, and IL-15 action. As we've discussed, ruxolitinib, tofacitinib, important when you're seeing JAK2 as a target, as in myeloproliferative diseases, but also pure JAK1 and pure JAK3 inhibitors that are a major focus of our attention. So in conclusion, our emerging understanding of the IL-15, IL-15R JAK-STAT system provides a definition of its actions, its disorders in disease, and opens distinct possibilities for the development of more rational immune interventions. These interventions could involve bringing IL-15 to therapeutic fruition in combination therapy for the treatment of cancer. And it could involve agents that block IL-15 JAK-STAT pathway for patients with select or organ-specific autoimmune diseases and with T-cell malignancy. Of course, one depends on a great number of others that actually do the experiments I discussed. Can I have that slide on? The next slide. So, as we've said at the beginning, attention must be paid to such a person as Bill Paul. I'm going to quote the words of those written at the bottom. Some scientists merely add threads to the tapestry of biology, but with Bill's passing, we have lost a master weaver who was brilliant at seeing the essential patterns in the fabric of nature. For his special capacity, for his leadership, for his friendship, he will be missed tremendously. Bill Paul, an extraordinary scientist, mentor, friend, and mensch. I second that entirely. Germain, Folks, Samuelson, and Lynn. Thank you for your attention.
We have time for a couple of questions, if anyone has any questions of Tom. That was magnificent. I don't see how anybody could have a question, but that was fantastic.
Thank you. Thank you. As I say, the second half was all new.
Thank you.
Thank you.